Today's presentation is FDA approval of an oral testosterone replacement enabled by Phoenix NLME and Trial Simulator. Today's speaker is Dr. Nastya Kassir. Nastya is currently a senior director at Certara's Integrated Drug Development Group. She joined Certara in 2008. Nastya is a scientific and regulatory expert for drug development programs for both small and large molecules, particularly in the therapeutic areas of pediatrics, orphan diseases, inflammation, and oncology. She has authored or co-authored more than 70 abstracts or manuscripts in the field of PK/PD modeling. Nastya earned her PharmD and PhD in pharmaceutical sciences from the University of Montreal in Quebec. She is a fellow of the American College of Clinical Pharmacology. Nastya, welcome to today's presentation. I'll now turn it over to her to begin the webinar.
Thank you, Susan, for the presentation. Sorry. Sorry, I'm having just some.
Just click on the deck to advance the slides.
Okay, it's not advancing.
Okay, I'll advance for you. Just tell me next slide.
Okay, thank you. So the field of modeling and simulation is transforming how drugs are developed and how they are regulated. In the FDA strategic plan on advancing regulatory science, 4 of the 8 science priority areas called for modeling and simulation. Before 1992, timelines of FDA drug review was a big concern. In 2017, the President signed the Prescription Drug User Fee Act that provides the FDA with the necessary resources to maintain a predictable and efficient review process until 2022. Among the other initiatives at the FDA was the launching of the Model-Informed Drug Development Pilot Program that provides drug developers and FDA an opportunity to discuss the application of MIDD approaches to their programs. The FDA would accept 2 to 4 applications per quarter. In addition, the FDA created modeling and simulation groups to support the expansion of this field and provide recommendations. Similar initiatives were also done in other countries, but we're just mentioning today FDA's initiatives. So the modeling and simulation are fast becoming indispensable across regulatory agencies. These strategies are helping to inform regulatory decisions, predict clinical and population-level outcomes, and protect the public health. Next. Earlier this year, Certara reported that more than 90% of novel drug approvals by FDA in 2019 were supported by Certara software or consulting services. Next. So now we will ask you to go to your smartphone or second monitor and to enter the 6-digit code into Menti to answer the questions, and I will hand it to Susan to help you with that.
Okay, everybody. As Nastya said, just go to menti.com and enter the code 799082. So we thought we'd just warm everybody up with an initial question, just get kind of light. So first question we wanted to ask you was, what's your favorite pizza topping? Do you like olives? Do you like pepperoni? Do you like pineapple? Are you like my kids and maybe you like plain cheese? Let us know what you like on your pizza. I'll just give everyone a minute to respond. Well, it looks like this is definitely a crowd that likes their meat. Pepperoni seems like the clear favorite, although more people than I would have thought like pineapple. So, well, I'm gonna move to the next question, but thanks everyone for letting us know what your thought on this important question is. On a more serious note, since this talk is going to be about both population pharmacokinetic analysis and trial simulation, we wanted to know, what tools are you using? Are you using Phoenix NLME for your pop PK analysis? Are you using non-mem? Maybe you're using Pumas or Julia or Monolix. Maybe you're using an open source tool like R. Let us know what you're using. Well, it looks like Phoenix NLME is pretty popular with this crowd. We're always happy to see that, and there's a good number of non-mem users, and we're happy that you've joined us today as well. So, thanks everyone. And next question is going to be on trial simulation. So, what tools are you using for trial simulation? Are you using Certara's trial simulator? Are you using non-mem or R? Maybe you're using some sort of open source tool or Simulo, or maybe you don't do trial simulation, and that's okay too, because that's why this is a great presentation for you to come to, so you can learn about trial simulation and how you can do it and how you can help use this approach to answer all sorts of complicated drug development questions. Well, it looks like we do have a couple trial simulator and R non-mem R users, but it looks like the bulk of today's audience isn't using trial simulation, and that's great to know, and it's great that you're here. Welcome. We hope that you find this presentation educational and you learn how trial simulation can really be a valuable tool. Nastya, I'm going to pass it back to you.
Thank you, Susan. Next slide, please. Okay, so today's presentation is about a work that we did for one of our sponsors, Clarus Therapeutics. So Clarus is a small pharmaceutical company that specializes in men's health. Clarus was developing an oral testosterone replacement product for use in the clinical management of men with testosterone deficiency. At the time when the sponsor contacted us, the phase 2 studies were completed, phase 3 study was ongoing. However, in the phase 3, the mean average concentration was higher than what they had observed in their phase 2 studies, and also the percentage of men with Cmax values, which is associated to safety concerns, were first higher than those observed in the phase 2, but also likely too high to be acceptable to the FDA. So consequently, the sponsor wished to conduct an additional phase 3 study with a new study design that will bring their average concentration and their maximum concentration down. In addition to these findings that we just mentioned, the FDA has mandated that a single blood collection point be identified that will function as a relatively accurate predictor of average concentration being in the normal range. Next. So the objective of this work was first to identify the time window for the single sample surrogate and to identify dosing adjustment guidelines for the new phase 3 study. And these objectives should permit assurance that the average concentration and maximum concentration levels are in agreement with the FDA guidelines for treatment in hypogonadal men. Next. So the PK efficacy and PK safety endpoints were presented in the FDA briefing document, and for the PK efficacy endpoint, it was mandated that at least 75% of patients should have their average concentration within 300 to 2,000 nanogram per deciliter, which is considered the normal range for average concentration. And for the PK safety endpoints, we had 3 endpoints to meet. So for at least 85% of subjects should have their maximum concentration below 1,500. Less than 5% of subjects should have their Cmax between 1,800 and 2,500, and 0% of subjects should have a Cmax above 2,500 nanogram per deciliter. So what can we modify in this design to answer the questions and the need of the sponsor? So first, does reducing or enlarging the threshold for dose adjustment improve the percentage of problematic Cmax, but still achieve average concentration within the normal range in more than 75% of subjects? And second, does changing the single sample, which is currently 4 to 6 hours post-dose, to another value improve the percentage of problematic Cmax? And finally, it's the titration approach. So does changing this titration approach, like start at the lowest dose and titrate upwards as needed, or start at the mid-range and titrate both upwards and downwards as needed, will improve our results? So these were the questions that we needed to answer for this work. So first, we needed to develop a population PK model based on the data we had from phase 2 and phase 3 studies, demonstrate that the model is reasonably accurate and predictive in order to perform simulations and modify the study design. So using Phoenix NLME, we started by building a base model using 2 phase 2 studies and the existing phase 3 data. We then performed covariate analysis to identify any important covariate in the model. So we checked weight, we looked at baseline testosterone of the patients, race, and also other covariates, in order to get a final model that was validated using visual predictive check and goodness of fit. And I say validated in order to be used for simulation to modify the study design using trial simulator. So the results of the population PK analysis of testosterone show that the testosterone is best described with a 1-compartment PK model with first-order absorption and lag time. An allometric function on clearance and volume driven by the body weight of patients was included in the model. And here we can see the graphical representation taken directly from Phoenix for this model. And we also have included between subject variability that describes how the concentrations vary from one individual to another, and also another component that's inter-occasion variability, which allows to describe how the concentrations vary from one visit to another in the same individual. The model was validated or was checked using visual predictive check, and as we can see, the 95% confidence intervals of the 5th, 50th, and 95th prediction percentiles overlaid with the corresponding percentiles of observed data. So the gray shaded areas are the predicted percentiles, and the line, the solid line, is the percentile of observed data. And what we noticed from this plot is that higher concentrations were robustly predicted, suggesting adequate prediction of Cmax, and then which is related to safety. Average concentrations were robustly predicted, suggesting adequate prediction of C average related to efficacy. But lower concentrations, so the fifth percentile, were associated to a higher uncertainty. But in our case, that was not a big issue because we were mainly interested in Cmax and C average. So here I wanted to show you the study original titration scheme from the original phase 3 study. And we can see that all subjects in the study started with a 200 milligram testosterone dose BID. On day 30, Day 30 is a PK day where testosterone levels were measured and titrations were made on day 42 depending on the identified range here, which was 250 to 1100 nanogram per deciliter. So if the concentrations were lower than 250, then the dose was increased to 300 milligrams, so a 100 milligram dosing step. And if the concentrations were higher than 1100, then dose was reduced by 100 milligram again step to 100 milligram BID. And those titrations were based on the sample collected at 4 to 6 hours post-AM dose. And then there was another occasion on day 60 where PK was collected again and titrations were made on day 74, and that was the last chance to make any titration before the end of study on day 90 or day 105. And we have used Trial Simulator to modify this titration scheme and to give better chances for the next study to meet the requirements by the FDA. Okay. So here I see that we cannot see all the slides I had, but it's okay. So I actually wanted to show how the model looked in Trial Simulator and how we can enter the parameters that were for the main PK parameters, the variabilities, the variance covariance matrix, and these were copied directly from Phoenix because the two softwares are compatible. The same illustration we had in Phoenix could be reproduced in Trial Simulator with the same parameters. And what we can see here is how the study design was written in Trial Simulator to allow for the adaptive design. So on the left, we see where we measure average concentration and on day 30, and we give the instruction what to do depending on the level of testosterone, and we ask the titration to happen on day 42. So exactly like the original design, and then down we see the second occasion for titration. Next. So the first thing after we have developed the model and we have it now in Trial Simulator was to verify that the model we have works well and that we can use it to change the study design. So the first step was to verify that our simulated or predicted concentrations are in agreement with the observed concentration first on day 30, so before any titration was done, only after taking so everyone is taking the same dose, 200 milligram BID, without any up or down titration. So we looked at the predicted proportions of Cmax and C average on day 30, and we know we have a good model because the frequency of the outlying Cmax within all categories, except for the above 2500, and also the outlying frequency of C average within or outside the normal range were accurately predicted with this proposed PK model. Next. And the next step was to, again, validate the model after the titrations were done. So we simulated, and that's at day 90. So we simulated a starting dose of 200 milligram. The single sample was taken between 4 to 6 hours to make the decisions for titration. The range for titration was 250 to 1100, and again, first titration step was 100 milligram, and the second titration step was a 50 milligram. And then we compared again the proportions of Cmax simulated against the observed ones, and we see that we were able to match the frequency within 1800 to 2500, and it's very similar to the observed. And then for the C average, we were able to have similar results within and outside the normal range. And you can notice that the frequency of maximum concentration above 2500 was much higher than the observed, but this at least gives us assurance that we are over-predicting and not under-predicting the concentration, and we are being on the conservative side for this work. So now that we have confirmed that we can use our PK model and that our simulation design can reproduce the observed results, we start looking at the different questions that we wanted to answer. So the first question was the impact of reducing or enlarging the titration range on efficacy and safety outcomes. And we can see that with the percentages we had in the original design, which is in green, and then the design that we selected, which is in light orange. So overall, reducing the 250 to 1100 bounds for titration to 250, 700 resulted in a marked improvement in safety with no change in efficacy. So safety was improved from 40% for the target for the less than 1500 to 57%, and for the efficacy endpoint, it was decreased a bit from 90% to 88%, but the target is more than 75%, so we are still meeting the target. And it appears that decreasing the upper bound, so to 700, is critical to improve the proportion of subjects that meet the safety objective but does not reduce the proportion of subjects meeting the efficacy objective. The next question was the impact of collecting time frame of the single sample. So, and the results here, again, so green is the reference and light orange is the results, the scenario we are proposing. And these results suggest that changing the 4 to 6 hour single sample collection time window to 3 to 5 hours can produce an improvement in the ability to move safety, which was increased from 40 to 53%, without affecting the likelihood of achieving the efficacy objective, which was decreased by less than 1%. And the last question in this work was looking at the impact of the starting dose level on reaching those endpoints. So decreasing the starting dose from 200 milligram BID to 250 BID resulted in only a slight improvement in the predicted ability to meet efficacy and a large improvement in the predicted ability to meet safety objectives. In contrast, in simulation 4A, a starting dose of 100 milligram yielded a substantial proportion of subjects with serum average concentration below the lower normal limit of 300, and we can see that this percentage dropped to 53%. So the next step was to combine all those simulations in an optimal design and do the simulation with all those changes. The proposed, so simulation 5C is the proposed scenario, which is 200 milligrams starting dose, 3 to 5 hours, sample for the single sample, titration range of 250 to 700, and 50 milligram steps for both occasions instead of 100 milligram step. And with this simulation, we were able to maintain a percentage of average concentration above the target, which is 75%, and to improve the percentage of subjects who meet the safety endpoint from 40 to 70%, even though it was not still ideal to meet the 85%, but there was a great improvement. And you can notice that in the previous slide, we said that 150 was better than 200 as a starting dose. But when all aspects of the design were combined, the 150 milligram starting dose provided average concentration below the normal, the percentage required for the normal range of average concentration, and it was 70%. And that's why the 200 milligram was used instead of 150 milligram. And again, here I wanted to share some snapshots from the trial simulator where we can see the different responses we can ask for. So in our case, I asked actually to see the average concentration on day 30. I wanted to check the average concentration on day 72, again on day 114. And we can specify when do we want those observation in addition to the other observations we had, like a clearance, Ka, lag time, the volume, and when did we want them to be measured. So here you can see that I asked for the concentrations to have a rich PK profile because I also wanted to confirm the results we were getting from the single sample. And we asked for the single sample to be drawn between 3 to 5 hours, and this is randomly drawn during that time frame. And then we can click on the time of a schedule of events to check that we are really getting what we asked for. So we get a big table with all the observations and when they are being scheduled to be sampled. Next. Here I wanted to highlight an important feature of Trial Simulator, which is creating scenarios. In the slide that we cannot completely see, so after defining the whole design, we can change properties of the design only by creating scenarios. So in this case, I was comparing, so my base design was a 200 milligram starting dose, and then I can just add here a property and ask for it to be changed, and then we can have all the simulation repeated by just changing one property here. So I will look at 150 or 100 milligram. But anything that was defined in the design, like the dose, the values of C average that we were using, the thresholds, the number of subjects, the number of groups, any property of the model can be added as a scenario and just modified here without touching the original design. And this is a very important feature that makes changing and comparing designs very easy. Next. So this is the key recommendations we had for the new phase 3 trial that the client had to run. As you can see, all subjects started at 200 milligram BID like the previous design, But now the range that prompted the physician to make a titration was changed to 250 to 700, and the dosing titration steps are now go from 200 to 250 or 200 to 250 in a 50 milligram steps to allow really for small changes and not, bring the subject one time with high concentration, one time very low concentration. And the sample, the titration were made on the sample collected at 3 to 5 hours post-dose. And then again, we had, as in the original design, we had another visit for another PK visit and a titration before the end of the study. And this new study design was used in the new phase 3 trial that the client had to run. And the ultimate, results from this work is when we get this letter, so that the FDA approved this new oral testosterone for treatment of men with certain forms of hypogonadism. So the take-home messages from today's webinar is that modeling and simulation is playing an increasingly important role in drug development and regulatory submissions. In our case, and in also other cases, Phoenix NLME and Trial Simulator supported the approval of this drug, and complex adaptive design features were tested by simulation to select the design with the best chance of success. So I want to thank the Clarus team today for their trust in us to support them during this process, and also my colleagues at Certara who either supported me for this presentation or doing the work. So thank you for your time today, and now we invite you to ask questions.
Thank you so much, Nastya. We encourage our audience to put their questions for Nastya into the Q&A box, and we will answer as many as we can. Looks like we've got our first question. Someone wants to know, how long does it usually take you to complete such POP-PK and trial simulation studies?
So it always depends. If there are like very tight timelines, we always try to to fit our work in those timelines. But in normal situations, I would say we can complete this work in 2 to 3 months.
Can you confirm how the average drug concentration was determined and why the sample window was considered a surrogate for the average drug concentration?
Yes, so the average concentration was actually determined using rich profiles. So when we mentioned the PK visits on day 30 and then again on day 72, I guess, so the sponsor and even us, when we did the simulations, we did a rich PK profiles. C average was determined from these profiles, and then after that, we looked at different samples that correlated best with these C average or the decisions made based on C average. And that's why we call it a surrogate of C average, because the C average was done on rich profiles, but then we had to find one sample that could give us similar results as if we have done rich profile, because this is not feasible later on when patients are taking the oral testosterone and they are supposed to monitor their levels, with time.
So someone wants you to comment on the following. In the Jatenzo, that's the branded name of the drug, in the Jatenzo label, the dose titration scheme was based on the 1 hour PK time point. Can you comment on that?
1 hour?
Maybe maybe the person needs to add some clarifying details. We can always get back to that person.
Yes, so I just want to mention something that, so we did not show all the details of the work that was done, obviously. So this was a very long procedure with time. As you can see, the client had to go back, run a study, and then, always working with the FDA on this. And then one of the things was also that after everything here we were presenting was like in milligram or testosterone, then they had to do the label using undecanoate testosterone. So you can see that the doses are slightly different because they had to express them differently. Yeah. So, but the 1 hour, I'm not sure, but I can check this and we can get back.
Yeah. The person clarified that the single hour was 5 hours post morning dose. So that clarifies things. Thank you. Someone else has a two-part question. One, they want to know what the result output formats are for Trial Simulator, and second, they want to know about training material for Trial Simulator. And Nastya can certainly comment on that, but I would mention that we conduct all of our training for Certara software products through Certara University, so you can go to our website and find information on training for Trial Simulator.
Okay, thank you, Suzanne. Sorry, what was the first part of the question?
They wanted you to comment on the result output formats for Trial Simulator.
Okay, so based on previous experience, we have submitted to the FDA the file that's used for simulation. In some cases, we have also provided a document that explains if we had multiple files, like what were the steps. But we did not submit like the outputs because they could be very large. But we submitted the file, the trial simulator file, and the input file where we had the characteristics of the population, and that's it. And then they can run it. The FDA has the software, and they are able to run themselves.
Someone states that testosterone has high variability. How did you manage the high coefficient of variability in your model?
It's right. It has very high variability. Frankly, I don't remember now exactly what were the variabilities in our model, but we were also able to see that it was not only between subject variability, but also there was an inter-occasion variability. And by taking in account those two parameters, we were able to have a good model, and like the visual predictive check were also in good agreement with the observed data. But it's true that variability is high, but sorry, I don't have the numbers now.
Someone wants to know, can you simulate a whole new PK profile for 24 hours with Trial Simulator and PK modeling without an actual clinical study if the conditions are known?
Yes, if there is an existing PK model in the literature, or even if we had previous data, so we can have a PK model and simulate with any conditions that we specify. But we cannot simulate without like any PK information.
What if you got results that were underestimated? What would you do in that case? As in this study, when you performed simulations, you got overestimations.
Yeah, then in that case, we would have been worried that, we might have in reality high concentration. We could have like more high concentration than what we are estimating. But then what would we have done? I guess in that case, you wouldn't be that sure about our model and continue the work. You would have gone back to the model, try to have a better prediction, or improve the model, look at other covariates. So look how we can improve our model before proceeding with the simulations.
Someone wants you to clarify, did the FDA approve the redesigned clinical trial plan, or did the FDA approve the drug without further clinical studies?
So the sponsor had to run the new study, and I guess at that time there were discussions with the FDA, and they ran the new phase 3 study, and after that they got the approval of the drug.
Looks like we have one final question from our audience. Do other regulatory agencies besides the FDA accept analyses done with Phoenix and Trial Simulator?
Yes, so our experience with the EMA, PMDA, and other agencies, we have submitted Phoenix and Trial Simulator files, and they were accepted.
Looks like I spoke too soon. So we've got another question came in. To add to the variability question, how did you handle endogenous baseline testosterone before dosing?
So in this study, the endogenous level was very low, but it was actually, it's just, let me remember, I think it was, so I know this was looked at. If I'm not mistaken, it wasn't taken in account just because everybody had a very low testosterone level at the entrance. But there were some models that looked at having the endogenous level in the model, but because the levels were so low at the beginning, this was not kept in the model.
Someone asked whether you considered modeling DHT rather than testosterone.
No, I don't think we looked at that.
Okay, great. Thank you so much, Nastya, and thank you so much to our audience for those excellent questions. I think we had some really great discussion here. Before concluding the webinar, we have a few short announcements. We are also co-sponsoring two upcoming webinars this month. This Thursday, Certara's doctors Mike Dodds, Samir Muksasi, and Yuan Zhang will present Model-Informed Drug Repurposing, Applications for COVID-19. This webinar will be presented by Pharmacometrics Africa. To register, visit pmxafrica.org. Finally, on May 27th, Certara's James Zoshak, Terry Ochiltree, Renata Levitch-Savi, and Robert Waterland will present Mitigation Strategies for U.S. and EU Supply Chain Risk Introduced by the COVID-19 Pandemic, and it will be presented by the American Association of Pharmaceutical Scientists. Visit aaps.org to register. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.